Hello, AP Bio kiddos. Welcome to the AP Bio exam review video series, Chapter 24, Early Life and the Diversification of Prokaryotes. You must know a scientific hypothesis about the origin of life on Earth. There's not a diagram that goes with this, but we're going to talk about the four different stages of hypotheses that are being investigated with regard to this one massive hypothesis about is it possible for life to come from nothing? So the four stages that we identified and that are in your book are first, the abiotic synthesis of amino acids and nitrogenous bases. So those are the monomers. Those are the monomers of our macromolecules. And so the first thing that would have to happen is that we would have to have that abiotically synthesized. We know how they're made in cells because it happens in our bodies all the time, but the question is, could it happen outside of cells? Once we have those amino acids, we'd have to join them together into macromolecules, and then we would have to package those into protocells. What protocells are is just a membrane that allows the interior environment to be unique from the exterior environment. That's all that a protocell is. It's just a membrane that maintains a different environment inside and outside. And once protocells are formed, then stage 4, the origin of self-replicating molecules that allow for inheritance. And so that's the last stage, having all those proteins that are necessary to replicate the macromolecule that is DNA. And so each of these four stages are being investigated independently in many different labs. Scientists have shown that for each of these four stages, that particular stage can happen in a lab, in a laboratory setting. And like we said in class, what that means is obviously subject to interpretation, and what scientists are trying to show is that if conditions were just so, is the phrase in quotes I put around, if conditions were just so, could it happen by itself? And nobody's put all four of these stages together to show that the whole thing can happen, but scientists are independently researching all four of those stages. You have to know the age of Earth and when prokaryotic and eukaryotic life are hypothesized to have emerged. So here it is, just written out for you. Quick summary. Earth formed 4.6 billion years ago, hypothesis by the Big Bang and the Big Bang Theory. And nothing happened for, what is that going to be, 0.8 billion years? So then nothing really, there's no evidence of anything for those first 800 million years. But then at 3.8 billion years ago, that's when we have our first chemical signatures of prokaryotic life. And then 3.5 billion years ago is when we get our first actual fossils of prokaryotic life. And remember, these things are dated by looking at chemical signatures in the rocks around the fossils, not in the fossils themselves. And then prokaryotes really had the planet to themselves for almost 2 billion years. 1.7 billion years is how long they were on their own. And then at 1.8 billion years ago, that's when we have our first fossil evidence of eukaryotes. So you have to know the characteristics of the early planet and its atmosphere. And so here's where we have to remember that oxygen in our atmosphere is coming from our plants. The plants are the things that are taking the carbon dioxide in and producing the oxygen as waste. So if we don't have eukaryotes, and plants are eukaryotes, then we don't have oxygen. And so as a result, we have an oxygen-free environment very early on in the planet's history. So that's what this is saying, very little oxygen. The atmosphere was also very thick with water vapor, lots and lots of water vapor. And then there were also all of these compounds that were released by volcanic eruptions. And so that's nitrogen, carbon dioxide, methane, ammonia, all that kind of stuff. And then as the Earth cooled, which took a very long time, the water vapor slowly condensed into water, and those formed our oceans. So to the question of that first stage, can we produce synthetically, abiotically, I should say, can we produce abiotically those monomers for macromolecules? That was the Operin-Haldane hypothesis, and the hypothesis was that it could have happened on Earth, like I said, if conditions were just so. And these two scientists, Miller and Urey, were the two guys who actually tested this operant haldane hypothesis. And this was their experimental setup. And what you're seeing here is a Bunsen burner. So we have heat. And this is going to be considered kind of like your oceans. It's producing water vapor. And added in here are a whole bunch of gases that are simulating the atmosphere of the Earth. And so this cycles around in this manner, just showing kind of the water cycle. The water cycle on the planet, where the sun comes in, heats up the water, it floats in the atmosphere, it falls down as rain, and so forth. They add in there sparks that are going to stimulate lightning. And then, as it condenses, it would come back around here, and they let this just go. The cycle just goes and goes and goes and goes and goes. And then every once in a while, they take a little sample out of the inside, and they see what's inside. And what they found was that this sample, after they let this go for a while, would actually contain amino acids and other organic compounds, not because they were put in over here, but because the materials that were in the atmosphere were assembled in such a way that amino acids could be formed. And this particular hypothesis, the operant haldane hypothesis, was modified by many different people who thought maybe the atmosphere had different stuff in it, maybe lightning would not have been as prevalent as we thought. And so they've used the same system with many different hypotheses to see if we could get amino acids out of it. And every time they run this, they're able to get amino acids into this little beaker here in the bottom. So what did they show? They showed that it is possible to abiotically produce monomers and other organic compounds from atmospheric gases. You need to know the key way in which prokaryotes differ from eukaryotes with respect to the genome, membrane-bound organelle, size, and reproduction. This is the best little table for this, and so I'm just going to copy and paste it in here, and I'm not going to read it to you because you can pause the video and read it yourself. But here are the biggies. Nuclear envelope, membrane-bound organelles. Those are the two huge things that you really need to know. Introns in genes. That's another one that you need to know. Circular chromosomes. Why can't I write? There we go. And histones associated with the DNA. These are the things that you really need to know. That's going to separate out your prokaryotes from your eukaryotes to help you understand what the differences are and how to interpret those things. So take a moment, pause the video, read through these things and make sure that you're good with them. It's not a difficult question, just make sure that you're remembering this stuff now that we are all the way into the spring. You have to know how the horizontal acquisition of genetic information occurs in prokaryotes via transformation, conjugation, and transduction. We looked at this in our last video, or maybe 2 videos ago, but we're going to look at it one more time. And so, because prokaryotes, they don't undergo mitosis. Remember, the mitosis, that's the phases where there's condensing and aligning and all that kind of jazz. Well, with prokaryotes, they just undergo binary fission. So their one circular chromosome pulls apart and they split in half. Their progeny is just a bud. And so in order for us to have genetic variation in prokaryotes, there have to be other ways for that variation to be introduced. And so one of the ways is through transduction. And so what's happening here is, as I said in the last video, our virus is coming in, it's injecting its DNA. That DNA hijacks the cell, the prokaryotic cell, and then that undergoes the self-assembly of the viruses, and then those split apart. But I want you to notice what happens in here. That particular virus, during the process of self-assembly, did not pick up viral DNA. Instead, it picked up prokaryotic DNA. But then that virus, because it's programmed to do so, is going to float around and find another prokaryote to try to infect. Only this time, instead of having the blue DNA that's going to repeat this process, it's got this brown chunk of DNA that is actually going to be contributed to another prokaryotic cell, and rather than undergoing this lytic cycle of bacteriophage infection, what it's going to do instead is inject that DNA, and crossing over will occur, not altogether too differently than how it happens in eukaryotes, and this recombinant cell is actually going to be reproduced. And so here, instead of the cell producing more viruses and whatnot, this cell is now a recombinant, one that has DNA from 2 different organisms. So here's what I need you to remember. I need you to remember that in eukaryotes, all of this genetic variation is introduced in the process of reproduction. You and I, we have the same DNA, more or less, that we had when we were born. And the reason I say more or less is because we have mutations that are happening in our bodies as we undergo cellular cell division. But for the most part, when we were born and what we are now are exactly the same. That is called vertical gene transmission. Here we're looking at horizontal. And what that means is that throughout the organism's life, here in a prokaryote, the DNA of that organism can change. Okay? And here it's happening via what's called transduction. It can also happen in the form of something called conjugation. And in conjugation, we have F plus cells and we have F minus cells. The plus cells, the F stands for fertility, and the plus cells are ones that can produce what's called a mating bridge. And when that mating bridge is formed, as that's this little thing right here, once that mating bridge is formed, that's when our plasmid can then go from one bacteria to the other, one prokaryote to the other. Again, horizontal gene transfer, okay? Because we're not looking at producing younger progeny, right? Because that's not how prokaryotes function. Well, what we're doing is, like I said in class, you shake hands and I give you some DNA and you give me some back. That is horizontal gene transfer. So here's conjugation, where they produce this mating bridge. The plasmid is replicated and then sent over. That's why we have a light blue and a dark blue strand. That mating bridge is broken, and now we have two F plus cells. Okay, conjugation. You must know how the previous mechanisms plus mutation contribute to genetic diversity in prokaryotes. So again, we've talked about all of those previous mechanisms, and that's where that genetic variation is coming from. But just to give you guys a little bit of math, the probability of a mutation in E. coli is about 1 in 10 million per cell division. So that's a really, really, really small number. However, oops, my exponents got lost. You have 2 times 10 to the 10th new E. coli cells in your body every day. So every single day, even though this is 1 in 10 million, seems like it should be a tiny number, every day you have 2,000 mutated E. coli in your gut, and that's per gene. Okay? There are about 4,000 genes in E. coli. So when we look at the whole organism, there's 9 million mutated E. coli per day per human host. So the sheer number of prokaryotes on the planet and just how many of them there are and how quickly they go through cell division, even though the rate of mutation, the rate is very low, it's 1 in 10 million, the actual numbers are so huge that there's a huge amount of genetic variation that is introduced just simply through the process of binary fission. So all of these things together is how we end up with such incredible variation with regard to prokaryotes. So that's it for the prokaryote chapter. I hope you're having a good day, and I'll see you in class.